Hello students, welcome back to Brilliant Education Doha, Qatar. So in this video session, I wanted to explain a small concept from the molecular basis of inheritance chapter that is called structure of nucleosome. What is that? Structure of nucleosome. Let me start with this small concept, okay? Now, structure of nucleosome. This is one of the important concept in molecular base of inheritance. And before we go to this topic, we all need to understand something. We have already seen in human DNA, 3.1 billions of nitrogenous base pairs are there. And in the structure of DNA, which was given by the Watson and Crick, in that DNA structure, we have seen the distance between the two consecutive base pair is 3.4 Armstrongs. If you calculate like this, you will get the total length of DNA is 2.2 meter long of DNA. So look at, let me take this is a cell, okay? This is cell and this is nucleus, and inside the nucleus is DNA. This is a DNA. Yes or no, students? How come this 2.2 meter long of DNA, 2.2 meter long of human DNA embedded, or this how come this 2.2 meter long of DNA is present in a tiny nucleus? That is possible because of a packaging of DNA inside the nucleus. I am a clear students. So today we are going to understand how this DNA is going to be present and how this DNA going to be present inside the tiny nucleus. What is the mechanism of this packaging? How the DNA is packaged inside the nucleus? Look at here. If you take the DNA out, this is called chromatin. Okay, what is this? This is called a chromatin fiber. Am I clear? Chromatin fiber. This DNA chromatin fiber during the cell division time, it started to condense. It started to condense to form a chromosome that we have already seen in the 11th grade. So look at here, this DNA started to condense and this DNA showing thicker, okay, like this structure, and in this thicker condensation, in between the DNA, we would identify some beads like structures are there. Look at here, some what are present here? Beads like this. Am I clear? This bead like structure or beads between the DNA, we can say nucleosome. Nucleosome. Am I clear, students? When DNA started to condense, okay, when DNA started to condense during the cell division time, it started in between the DNA, there are the small bead-like structures are there. The bead-like structures are called what? Nucleosome. This nucleosome made up of two things. One is called DNA. Second one is called histone protein. What is the second one? Tell me. Histone protein. Everyone keep in your mind. Histone protein, we can also say histone octamer, and around this histone octamer DNA will be wrapped. Am I clear? Today I will explain you how this total DNA is wrapped around this DNA. Let me discuss with this histone protein. Histone protein, we can also say it is octamer. Octamer. What is this octamer here? In histone protein, there are, look at here, in histone protein, there are 8 protein molecules are there. Look at here. This is H2A, okay, H2B, this one is called H3, H4, okay. These are histone octamer proteins, each one in the form of 2, okay. This is H2A. Now this is second H2A, second H2B, H3 and H4. Am I clear? So there are total 8 ball-like structure of protein molecules are present, and these 8 ball-like proteins are called histone octamer. But wait a moment, here only proteins are there. The nucleosome is made up of DNA plus histone, right? Look at here. So histone octamer. Now all of you look at here. Around this histone octamer, DNA comes and wrap. Look at here. DNA will be wrapped around this nucleosome, or we can say histone. Am I clear? Like this. I hope you get my point. Am I clear? So this total structure, we can say it is a histone octamer. Second one, this one we can say it is a DNA. Am I clear? So like that, the DNA will be wrapped around the histone proteins. Look at here, like this. So let me take a DNA here. This is the DNA and these are the histone octamers. Okay, and it will be wrapped around, come down. Again, here will be one more histone. Okay, again it will be wrapped around, and like this, the DNA will be wrapped around the histone proteins. And histone proteins are rich in two different type of amino acids. Histone proteins are rich in two different type, or rich in the amino acids called lysine, and one more amino acid is called arginine. These two amino acids we have already seen in the biomolecule chapter 11th grade. These two amino acids are basic amino acid. Yes or no? So these two are basic amino acid. That means I can tell you histone proteins are negatively charged. Okay, what is the charge? Negatively charged and basic in nature. Am I clear? Now, basic and negatively charged histone protein around positively charged acidic DNA will be wrapped. Am I clear, students? That's how they started to condense to form finally chromosome. During the cell division time, the condensation of DNA finally leads to the formation of, tell me what? Chromosome, like this. Am I clear, students? Now, keep in your mind, the package of DNA inside the nucleus is done because of the DNA is wrapped around where? Wrapped around where? Histone proteins. Now, if you look at this structure, there are two things are there. This is a DNA which wrap around the histone protein. That DNA which we call core DNA. The DNA which wrap around the histone octamer we call the core DNA. One more. Here, one more nucleosome, one more histone protein is there. Yes or no, student? Here, one more histone is there. Let me write here. This is H2A. Okay, this is H2B, H3 and H4. There are how many? Two are there. Yes or no? Two are there. And now this DNA again will be wrapped around. Coming. Okay. Now look at here. This is also DNA. Here look at this is also DNA here. Now students, keep in your mind the DNA which present between the one nucleosome to another nucleosome. This DNA is known as linker DNA. Which DNA? Tell me. Linker DNA. So this total structure I can tell you now. Okay. This total structure I can tell you now. Nucleosome. Nucleosome. This is also what? Nucleosome. Nucleosome. So between the two nucleosome, whatever the DNA is there, that DNA called the linker DNA, and the DNA which wrap around the histone octamer we known as core DNA. Am I clear, students? Now inside the nucleus there are two different type of, that mean few DNA, that means little amount of DNA is darkly strained, tightly packed. Again, once again, I'm telling you, in the DNA, in the nucleus, some amount of DNA which is darkly strained, and tightly packed. So that DNA we can say heterochromatin. Heterochromatin. Remember, heterochromatin is that DNA part which is transcription wise less active or inactive, we can say. Okay, so the DNA which inside the nucleus lightly strained and loosely packed, that DNA we can say euchromatin. So let me here write what are heterochromatin. First one is called heterochromatin. chromatin, heterochromatin. Second one is called what? Tell me. Euchromatin. Chromatin. So first one is called heterochromatin. Heterochromatin is that chromatin. It is darkly strained and it present tightly. Am I clear? Tightly. And transcription wise, if you look at transcriptionally inactive. It is inactive. Many times in the examination question have been asked the difference between these two type of what? Chromatin. One is called euchromatin, one is called heterochromatin. Heterochromatin is the characteristic feature like this. And what about the euchromatin? It is lightly strained and loosely present, loosely arranged. And this DNA is transcription wise, transcriptionally, yes, transcriptionally active. This is what? Transcriptionally active. Remember, euchromatin and heterochromatin. So students, I hope you understand what exactly structure of nucleosome. Nucleosome is made up of DNA and also histone protein. But a last point. Sir, how these all 8 ball-like structure that DNA, the histone proteins are held together? There is one more protein is there which will hold this all 8 histone octamer proteins that we can say that is called H1. One more holding protein is here. This is called the H1 protein. H1 protein will not going to be inherited. So this is H1 protein. Am I clear? Now look at here. H1 protein function is just it hold. Am I clear? Just it hold the 8 histone octamer in a proper position. Am I clear? So students, remember nucleosome structure. Nucleosome is made up of DNA and also histone octamer. Histone octamer having the H2A, H2B, H3, H4, 4 plus 4. So total 8 proteins are there. So DNA wrap around this 8 protein. That's how the 2.2 meter long of DNA is present in a tiny nucleus. And this chromatin fiber started to condense to form chromosome during cell division time. So many times question have been asked me about the euchromatin, heterochromatin, nucleosome. Nucleosome is positively charged, negatively charged. How many amino acids are there? What type of amino acids are there? Okay. So important MCQs also in this one. So try to revise once again before the exam. Thanks for watching.